in this screencast, we are going to have a look at the process of photosynthesis. On this photograph on the screen, you can see close up of some plant cells, and in this particular case, they are plant cells of moss, and you might recognize seeing something similar in core biology practical we did earlier in the year. The oval shapes inside the cells are chloroplasts, and they contain this particular type of chemical called chlorophyll, which is really important for photosynthesis. So on the next slide or so, I'm just going to have a quick reminder about something we already covered in core biology, and specifically its reaction which summarizes photosynthesis. So you should be able to remember that photosynthesis is done by types of organisms called autotrophs, and what they're able to do is take carbon dioxide, which is CO2, combine it with water or H2O, and produce glucose, which is C6H12O6, and oxygen, which is O2. And another reminder is that I can't just leave this equation in its current form, because it would be an unbalanced equation, because on this side, for example, for carbon I have only one atom of carbon, which is on the other side I have 6 of them, so that would not be equal number. So in order to resolve it, I need to put 6 in front of every chemical apart from glucose, and this will result in balanced equation of photosynthesis. So if we now look in more detail at a chloroplast, we will see that it contains a number of different structures. So first of all, it's got double membrane. There is inner and outer membrane. And then there is cavity, which is called a stroma, which is filled with fluid. And in stroma, you have this series of discs called thylakoids, and they're stacked into structures called granum. So it's shown here. Granum is a single stack of thylakoids, and then grana are many stacks of thylakoids. So if we now take a single granum and look at it in more detail, then you will see that this granum, in this particular case, is composed of 3 thylakoid discs. In reality, there could be a lot more. This is just to represent the idea. And these discs, if you sort of do a cross-section of them, then inside there is thylakoid space, again filled with fluid, and the wall of thylakoid, the membrane is essentially phospholipid bilayer, which is again something we talked about in the core biology module. And once in a while, in this phospholipid bilayer, you have these specialized proteins which contain lots of molecules of chlorophyll. So on the diagram, they're highlighted in this dark green color. So chlorophyll, as I mentioned earlier, it's a type of molecule, and it's able to absorb blue and red wavelength of light from the sun, and is able to capture this light and convert it into chemical energy. And we're going to talk about that in a few minutes. So blue and red light gets absorbed, gets converted, by chlorophyll, whereas green light gets reflected, and that's why plants appear green to our eyes, because that's the bit which reflects back and is captured by our retina. So photosynthesis has got two main stages. The first stage is known as light dependent. As the name suggests, it requires the energy which comes from the sunlight. And then the second stage is known as light independent, sometimes in some books also known as dark, um, stage or dark reactions. Though it's not sort of particularly accurate way of describing these reactions, because it's not like they need to happen in darkness at night, for example. They can happen during the day. However, they do not need sunlight to happen, and that's why they're called light independent. So in terms of where these two stages happen, the first stage, the light dependent reactions, they happen within thylakoids in granum or grana. And then the second stage, the light independent reactions, they occur within stroma, within that sort of cavity which is filled with fluid. So just to summarize the idea behind those two stages, I guess you could describe that the aim of the first step, the light dependent step, is to sort of take energy from the sun and capture this energy within the energy storing molecules. And these two energy storing molecules, they're called ATP, which is something we'll hopefully remember from core biology, and another molecule known as NADPH, or also known as reduce NADP in some books. So basically you're taking this energy from the sun and holding on to it and converting it into chemical energy of these two molecules. Then once you've done that, then these molecules ultimately will be used in the second step of photosynthesis to produce carbohydrates from carbon dioxide. So this is sort of what happens within the first and second stage in a nutshell. So this is a diagram which summarizes these two stages of what we're kind of looking at. So the first stage, which is I've put in the red box, is the light-dependent reactions within photosynthesis. So here you're going to capture that light using chlorophyll. You're going to produce your two molecules, which is ATP and reduced NADP. And also you're going to feed some water into the system and you're going to produce some oxygen. So we're going to look at this stage in more detail next. So this is a little summary of light-dependent reactions which I kind of produced. I drew my own diagram in the end. Now, in reality, this process is extremely complicated. So even though the diagram might look complicated, but I promise you this is just a simplified version of it. And this diagram is there just to kind of capture the gist of the main processes which happen within that first stage. So let's start here. So here we've got chlorophyll, and this is, you remember, embedded in the membrane of this thylakoid membrane, thylakoids. So this is where this whole process occurs. So light is gonna get captured by chlorophyll, and chlorophyll is a type of molecule, which means it's made out of atoms. And atoms in turn contain small particles called electrons. You maybe again recall some of it was kind of covered in core biology briefly. So as chlorophyll captures this light, some of the electrons within chlorophyll are going to get excited. So that's why I put them in red, to show they're excited. And that means the energy level is going to increase. So it goes from lower energy level to higher energy level. And as these electrons are going down in energy level, the energy which is released is going to be used to produce ATP, which is what that bit suggests. And again, you might recall that in order to produce ATP, you need to take ADP, diphosphate, and add another phosphate to it to produce ATP, adenosine triphosphate. So that's what happens here. So in essence, you've captured that first bit of light energy by exciting electron and then using the energy of that electron coming back down from its excited state to produce ATP. Once that happens, then that electron is going to absorb another portion of light and again get excited, and you can see that that excitement energy state kind of became even higher than it was in the first instance. So it got excited again, and then as it's coming again down from its excited state, that energy is again going to get captured and it's going to end up producing this substance called NADPH. And you do it by taking NADP and then using electron to kind of combine it with that substance, and you're also going to need ion of hydrogen or H plus to be able to produce that sort of resulting substance. This substance is also sometimes known as electron acceptor because it sort of captures that electron. So just to sum up, right, so you excited electron once, produce ATP, excited it again by using light, another portion of light, and then produce that second substance. And those two substances essentially allowed you to capture a little bit of energy which came from the light and kind of conserve it, a bit like the little rechargeable batteries. Remember that analogy I used before. So kind of recharged those little molecules, and now you can use this energy to transport it somewhere else within the cell or within chloroplast and produce then carbohydrates using this energy. Now, if you kind of go back to the beginning of this step, you remember chlorophyll had this electron or electrons which got excited, and this electron is actually going to leave chlorophyll altogether because ultimately they're going to end up tied down in the substance which is NADP. So in order for chlorophyll to go through another kind of cycle of getting electrons which are excited, it needs to replenish electrons to start with. So where is this electron or electrons are going to come from? And they are going to come from a process called photolysis. And this is a process where plants take water, and again they use sunlight to sort of power this process, but essentially they're able to take water and split it into oxygen and hydrogen. So oxygen just becomes O2, and plants then release that oxygen. But hydrogen kind of splits further into electrons and into kind of the nuclei basically, or on in terms of chemical formula, it's H plus. So electron is going to go and rejoin chlorophyll and kind of replenish the electron or electrons which were lost, whereas hydrogen ion is going to actually go all the way to that reaction here to produce that substance NADPH. Okay, so it sort of looks pretty complicated, and I can appreciate that. So I actually found this little animation which hopefully will help you to understand it a bit further. So this is light-dependent reactions analogy, which you can find on BioNinja website, and there's going to be a link to it on the PowerPoint. So let's have a look at it together. So this is the electron which is in chlorophyll, and we've got this imaginary guy with a hammer, which is going to absorb a bit of light, and that's going to power the hammer to lift this electron and shift it all the way to the top of the energy tower. So then this electron, as it's coming down with its energy, that's powering a little pump, which is like an enzyme essentially, and this enzyme is going to produce ATP. Then we've got another chap with a hammer, which is going to capture another bit of sunlight, and again this electron then jumps up the energy level, and this time is going to get captured by this substance NADP. So then as a result you produced ATP, you produced NADPH, and then now all you need to do is just replenish that electron which was lost by chlorophyll, and you're going to do it by splitting water into electron, oxygen, and also hydrogen ion or proton, which is actually not shown on the animation, but in reality it's going to join that NADPH substance. And that's kind of the little summary of this process. So the whole thing looks a little bit like letter Z, which is on its side. So sometimes these light-dependent reactions are known as Z reactions or Z scheme. So you might come across that. And that's where that term comes from. Okay, so let's go back to the PowerPoint. So that this bit was the summary of light-dependent reactions. So then we need to look at the second step, which is light-independent reactions. So these reactions do not require sunlight. They can happen, however, during the day or during the night in the plant. And in essence, this is where you are going to take carbon dioxide, and you're going to use energy which we captured in those two molecules to produce glucose, or it could be other types of carbohydrates. And this stage is happening in stroma in chloroplast, as you can see. So again, these reactions in reality are extremely complicated, and this is just a quick summary of what happens. These light-independent reactions are also known as Calvin cycle, named after a scientist who kind of described and discovered these cascades of reactions first. So in essence, what happens is you're going to take carbon, a single carbon from carbon dioxide, which is what this white circle is, to represent a single carbon in CO2. And you're going to combine it with a type of sugar called ribulose biphosphate, which contains 5 carbons. It's 5 plus 1 equals 6. And this 6 carbon compound is very unstable, and it's very quickly breaks up into 2 lots of 3 carbon compounds. So you're going to end up with 2 lots of this compound called GP, glycerate phosphate. So that's what these sort of 3 circles represent, 3 carbons within this GP compound. So this is the first step of Calvin cycle, called carbon fixation, because you're capturing that carbon dioxide and fixing that extra carbon atom and feeding it into the cycle. Then the second step is we're going to use that energy we captured earlier from ATP and reduced NADP, and we're going to power the second step of a process where we're going to convert this 3 carbon compound into a different 3 carbon compound called GALP, or glycer. Let me try. I'll have a go at pronouncing it. Glyceraldehyde phosphate. Yeah, I'm sure you can do better than me. So this compound is really the one which the photosynthesis is really all about. So this is the compound which is then used as a basis for producing glucose. So some of this compound is going to be taken out of the cycle to produce glucose, and some of it is going to stay within the cycle and be kind of converted back into this sugar, which was ribulose biphosphate. And in order to do that, you need another portion of energy, which came from ATP, which we produced during light-dependent reactions. So here we are. So we've got your carbon dioxide getting combined with sugar, then being split into 3 carbon compound, then converted into another 3 carbon compound, and then some of it is going to kind of power the cycle again and become ribulose biphosphate, and some of it will become glucose. Now, one interesting thing about this cycle is this enzyme, which is called rubisco. And this enzyme is the one which powers the first step of Calvin cycle. We're going to take carbon dioxide and combine it with ribulose. And this enzyme is considered to be the most abundant enzyme in nature. So if you take soluble protein found in leaves, around about half of all protein in leaves comes from this one type of enzyme. So you can realize sort of that it is extremely important substance and extremely important type of protein in plants, or more generally on planet Earth. So this is the kind of summary of a process. Now, I don't want you to be kind of worried about the complexity of the process, or kind of the aim is not for you to memorize it all. The aim is kind of to appreciate just how complex some of these chemical changes are. And something as simple as we summarized in the equation of photosynthesis, where it just shows you, well, all you do is just combine a bit of carbon dioxide with water, and there you go, this is your glucose. In reality, behind this equation, there is a lot of really complicated chemistry. And what I described in this video is actually a very much a simplified version of that. So when you go onto your degrees, if you're doing biology or biomedical sciences, it's very likely that you're going to look at this process in a lot more detail. And just to finish things off, one more slide to those of you who are interested in photosynthesis, and kind of to link this topic back to core biology. So I just wanted to show you some pictures of what some of these molecules we talked about might look like. Obviously, these are just sort of representations of these molecules. In reality, I hope you remember that molecules, even the largest ones, they can't be seen directly under the microscope. So these are sort of our models based on our best understanding of the structure of these molecules. So this picture, first of all, it shows you structural formula or structural model of chlorophyll. So each circle is an individual atom. So the green circle in the middle is actually an atom of magnesium, and then the blue circles are atoms of nitrogen. So if you think about plant food, for example, if you want your plants to grow well, a lot of plant food and fertilizers in general, they contain magnesium and nitrogen, and this is in order for the plants to be able to produce plenty of chlorophyll, amongst sort of other things. Then this diagram here, if we were in class, I'd probably ask you, what do you think this is a picture of? But I'm going to just simply reveal that this is the enzyme. How do I know it's enzyme? Because it's a globular protein, which is what majority of enzymes are. Globular simply means like round in shape. And this particular enzyme is the Rubisco enzyme, which we need for the Calvin cycle. Now, these two pictures are not on the same scale. So something like enzyme is a relatively large molecule, whereas chlorophyll is going to be much smaller. But this is just to kind of give you an idea of just complexity of some of these molecules and substances involved in the process of photosynthesis.